Translation is the process of converting messenger RNA into protein. It all starts with the gene that codes for the protein or the polypeptide chain. So the whole process of transcription has controls and regulation, and then after transcription occurs where you have a messenger RNA, there can be further regulation and modification. So there's specifically with messenger RNA, adding a 5' cap and the poly A tail, and then RNA splicing that would go on, and then that messenger RNA is transported out of the nucleus into the cytoplasm where translation can occur. So translation is the process of having this messenger RNA, attaching some ribosome to it, and then using transfer RNAs to attach amino acids to the growing polypeptide chain. So the three main kind of general steps are initiation of the process, elongation of the polypeptide, and then termination of the process. So the transfer RNA is just a kind of it's a way to have the complementary RNA be able to detect the RNA sequence on the messenger RNA, and at the same time bring with it, for that specific sequence of RNA, a specific amino acid. And then the ribosome is kind of a complex piece of RNA and protein. Again, I think some of the RNA in it might be needed to bind to the transfer RNA or detect the RNA in the transfer RNA. RNA in the ribosome can find RNA in the transfer RNA to kind of help. I don't know. Maybe that's not correct, but either the RNA sequences in the ribosome play a role, or RNA in the ribosome is just strictly like an enzymatic kind of activity of providing form and function to the ribosome. But the messenger RNA has the codons, and then the transfer RNA has the anticodons. And there's degeneracy in that. So the same few codons can attach to the same transfer RNA. So in this case, the third codon is kind of a kind of an unspecific codon, where the first two have to be A in this case, and the last one has to be a pyrimidine, either urine or cytosine. And that's considered kind of base pair wobble, where that last base pair, I think that's what this is. The last base pair kind of can wobble between a few different scenarios. So, and I guess in other words, there's roughly 64 different codons if you include stop and start. We'll start usually as methionine, but there's say 61 codons, but there's a lot fewer transfer RNAs that actually attach to those. Um, so, in order to kind of start translation, um, the um, the ribosome's kind of finding this start site before it's going to start appending amino acids. So there's a frame that it's looking for. You can see here this different frames that are available. So there's 2 strands of DNA. There's 1 strand of messenger RNA, and so there's 3 different frames on it. I guess this page is overlapping genes, so this is at the DNA level. At the DNA level, there's 6 different frames, 3 on one strand and 3 on the other strand going anti-parallel. So I guess this should have been in the transcription section because it's about genes, but you can see that there's overlapping, especially in viruses and prokaryotes. There's overlapping frames, reading frames. So you can express multiple proteins off the same messenger RNA just by starting at a different frame. And in eukaryotes, it's back at the DNA level, but there's overlap, and it's not on the same strand, so it's not on the same messenger RNA, but it's on, there'll be 1 protein on one DNA strand and then a different protein on the other DNA strand, possibly frame shifted. So just like transcription, there's post-translational modification and post-translational regulation. So the modification is adding a phosphate or other functional group to it, or maybe related to information on transport that needs to be removed from the polypeptide chain before it's an actual functional protein. And then the regulation side of it is, the proteins can be immediately degraded if they're not needed. And then maybe the modification is also considered a regulation. So the chain as it's being created by the ribosome is more of a polypeptide until it's long enough or starting to fold. Then it's considered a protein. So I think the arbitrary cutoff is like 50 amino acids before it's considered a protein. And then as it's going through translation, a lot of times it'll start folding before it's completely translated.